Well, aloha everyone, and welcome to Unit 4 of Biology 101 here at Chaminade University. Today we'll be talking about cell structure and function. We're going to start discussing cell theory. Basically, the cell theory is that all living things, or even the smallest living things, are comprised of cells. We'll talk about the basic attributes of cells, so what makes a cell alive. And also we'll talk about the major differences between eukaryotic and prokaryotic cells and also some of the similarities. So what is a cell theory? The cell theory basically says that cells are the basic units of life, and the major principles of cell theory are that number one, every living organism on Earth is comprised of one or more cells. Now again, prokaryotic organisms would only have one cell, and multicellular organisms would be eukaryotic organisms, so they would have more than one cell working together. Again, the smallest organisms are unicellular or prokaryotes, and multicellular organisms are broken down into the smallest functional units of cells. Last but not least, all cells arise from pre-existing cells, and a little subset of this is that like cells arise from like cells. So what that means is just like a mother dog would never give rise to a cat as offspring, certain cells that have already gone through their cell lineage, like a heart cell that's going to create another cell, would create another heart cell. It would not create an eye cell. So once cells have gone down their differentiation lineage, they can create more cells, but only of their existing cell line. So not only do all cells arise from pre-existing cells, but they arise from cells that were similar to themselves. All right, so lots of different sizes of cells. Actually, they range in size from approximately 1 to 100 micrometers in diameter, which I know that doesn't sound like it's very large, but if you picture that's a factor of 100-fold, you can picture that some cells are much, much larger than others. But they don't get any larger than 100 micrometers in diameter because of the rate of diffusion. So as we mentioned previously, diffusion is how molecules are going to move into or out of a cell, right? From a highly concentrated region to a lesser concentrated region. And if you figure that a cell gets way too large, you can picture like an apartment complex, right? If the apartment complex continued to grow, then the people on the very inside of the apartment complex that lived in apartments would not have any windows. They might not have any access, or it would be very difficult for them to access the outside, and they might end up suffocating. And the same thing would happen with the cell. It would end up suffocating in its own cellular waste and not able to get enough oxygen, so it would end up oxygen starved, because cells need to exchange nutrients and waste with the environment just like a human individual does. And for that reason, that no part of the cell can be too far away from that external environment where gas exchange and nutrient exchange occurs. And so cells, as they start to get too large, they're going to divide to get a little bit smaller. Now, for the purposes of this class, we're going to be staying mainly within here, within the range of we'll talk about DNA and proteins, and we'll get up to eukaryotic cells and eventually to adults, but we're not going to end up in the extreme sizes. But if you want to compare, for example, the different sizes of something like DNA, which is going to be compared to or a little bit larger than something like an atom, but is going to be much smaller than something like a virus. Now, a virus is going to be the very largest thing that is going to be considered not alive, right? As soon as we get into prokaryotic cells, that's going to be the differentiation between a virus and well, life and non-life. And why is a virus not considered alive? Now, this isn't going to be on the test, but a virus is not considered to be alive because a virus is unable to replicate itself without hijacking host machinery. So while it contains all of the DNA or RNA, depending on what the virus is, to be able to create more virus, it does not have its own cell to be able to use all the cellular mechanisms required to be able to create more virus. So although it is able to make more of itself, it only has the ability to hijack other machinery from another cell. So it's not considered to be alive because it by itself is not able to reproduce. As we move forward here, we see most prokaryotic cells are on the order of under 1 micrometer. Interestingly enough, here's the mitochondria, which is considered to be an organelle, but there's actually new information that it might be actually an organism as well. So it's kind of in that gray area of science where the leading forefront is kind of challenging the old notion. But anyway, mitochondria are still considered to be organelles, and they are found inside most eukaryotic cells, both plant and animal cells, as an organelle. And here's where we're finding most of the cells that we consider to be eukaryotic cells, approximately 10 micrometers to 100 micrometers. The very smallest one would be a red blood cell, which would be 7 micrometers in diameter. But interestingly enough, a red blood cell doesn't have a nucleus, which means it doesn't have any DNA, and it also doesn't reproduce on its own. So similar to a virus, it doesn't have the ability to create another red blood cell. And so it actually, even though it is considered a body cell, it doesn't really fall into the same classifications as the rest of the eukaryotic cells because it is what we call anucleatic or lacking a nucleus. Moving forward, this is an embryo of a frog. It's approximately 1 millimeter. You can see that it's comprised of multiple different cells. So as cells combine into a multicellular organism, the organism is going to get larger and larger. This here is a chicken egg, which is going to end up either becoming a chicken if it is fertilized or end up providing albumin to other organisms. It's actually something that we use quite often as a source of protein, for example, because it's high in, as I mentioned, albumin. And this figure is also depicted by, separated out by what's able to be seen with a light microscope, what needs to have special microscope like an electron microscope in order to see things like atoms or DNA or proteins, and what's visible with the human eye, which is approximately the embryo level and up. All right, so what are the common features of cells? First and foremost, they have a plasma membrane. This is important. This is like having the roof and the walls for your building. This is what keeps the inside separate from the outside, which allows us to have, again, homeostasis or homeostatic conditions on the inside of the cell. Inside the cell, we also have cytoplasm. Cytoplasm is going to have cytosol, which is the liquid portion, and the organelles, which are going to be more solid. They're membrane bound generally. And this is going to be a difference between prokaryotes and eukaryotes. We'll talk about that in just a second. But either way, both of them are going to have a cytoplasm. And they all use DNA as a hereditary blueprint. That means that DNA is what gets copied from mother to daughter, right? From one cell to the offspring of that cell. And RNA is what's used as a copy of that DNA to be able to copy the blueprint and guide construction of the cell parts. And basically the central dogma here is DNA creates RNA creates protein. And you can kind of think of this as like if the DNA is an old recipe book that has every recipe for every kind of food you can possibly imagine. And then you really only want to make one particular thing. You want to make a cherry cobbler. So the RNA is going to make just a Xerox of the particular recipe that you want, that cherry cobbler recipe. So now it's a black and white Xerox, and it's allowed to leave the nucleus, and you're able to use it to construct proteins, which would be, in our example, the cherry cobbler itself. So the RNA is basically a copy of the blueprint that's used to guide the construction of whatever it is that we're creating, which generally is going to be different proteins. So let's talk about the plasma membrane for a second. The plasma membrane is very important because it is what separates the cell from the external environment. And this is what it's semi-permeable, so it allows interactions between the cell and its environment, but it regulates, right, what comes in and what goes out. And it can change what's allowed in or out based on what it needs and what it has excess of. The structure of the plasma membrane is mainly those phospholipids that we talked about previously, but also has a lot of proteins that are embedded in them that might have specific functions like receptor proteins, etc. And also carbohydrate molecules, so different glucose molecules might be inside there. And also that's going to be important in regulating molecules and ions into and out of the cell. So this is an overview of the plasma membrane. And you can see the plasma membrane is mainly comprised of what we call a phospholipid. Now you see the word bilayer, and that's because the phospholipid bilayer is actually one layer of phospholipids on this side and one layer on that side. And as we mentioned previously in Unit 4 when we talked about biological molecules, phospholipids have one region that is hydrophilic, or likes interacting with water, and one region that is hydrophobic. And this allows it to interact with the other layer, which also has the hydrophobic region, and then allows us to have a separation of two aqueous environments. So these heads on this side interact with this aqueous environment, these heads on that side interact with that aqueous environment, but it allows us to help keep the inside separate from the outside. Now, it looks as though these are all kind of static, but it's actually a fluid mosaic model. Basically what that means is that any one of these phospholipids can move about wherever it chooses to, and additionally, these guys can kind of move about as well, so anything embedded in the membrane can collect together or segregate apart, however it chooses. And inside this membrane, we have several things embedded. First, we have proteins. Proteins can be channel proteins that allow things in or out, or carrier proteins, same thing, but it can also help in cellular communication with like the external environment, for example. It could, for example, be a receptor for something like a endorphin that might make you have a fight or flight response. Additionally, we have carbohydrate molecules in here. These are called glycoproteins. These proteins are actually identification markers that say what type of cell it is, what type of organism it is. So not just that it's a pig cell, it's a pig heart cell, or not just that it's a human heart cell, it's Dr. Griffith's human heart cell. So very specific identification factors, and this is one of the reasons why people who get transplanted organs have to take immunorejection drugs, because that organ comes with a specialized glycoprotein that is not self, which means that the individual might attack it if it doesn't take the the anti-rejection drugs. But I digress. Some of the other things that interact here with this plasma membrane include these cytoskeleton molecules, and this is the scaffolding or the structural proteins that are going to help keep the cell structurally sound. All right, so let's talk about each of these individually. Inside the cell, we have the cytoplasm. Again, the cytoplasm is going to have the fluid part, that's the cytosol. That includes water, obviously it's an aqueous environment, but also salts and other organic molecules that might be dissolved inside that cytoplasm, like sugars, etc. And this is where all of the metabolic activities of the cell are going to occur. Again, this is where protein synthesis is going to happen, where proteins are going to interact with other proteins, where all of the biomolecules are going to interact with each other in the cytoplasm. Now, this here is an animal cell, as opposed to, I'm going to step forward one, this here is a plant cell. Let me go backwards. This is the animal cell. How do I know it's an animal cell versus a plant cell? Couple major differences. Number one, you see this flagella. A flagella is specific to animal cells, so are cilia. You're not going to see that in a plant cell. Secondarily, you can see this is kind of a oval shape, right, as opposed to a brick shape, and that's because it has a cell membrane, but it does not have a cell wall. Lastly, we're lacking a specialized region called chloroplasts. Chloroplasts they would be depicted like a little green stack of pennies perhaps, and chloroplasts are a specialized organelle that animal cells do not have, that plant cells do. All right, so let me move forward to the plant cells so I can show you those differences. Oh, also there's a central vacuole that is only found in plant cells. So if you were given an image and you had to determine whether or not it was a plant cell or an animal cell, the very first thing that would be a no-brainer would be, does it kind of look more like a brick? And that's because the plant cells are going to have this cell wall that surrounds it. So not just the cell membrane, which still exists in here, just to be clear, the plasma membrane, but it also has a cell wall surrounding it. Additionally, it has these specialized structures called chloroplasts. We'll talk about each of the specialized structures individually. And it has a central vacuole, which again plays the same role. Other than that, it has a lot of the same organelles. So I'm going to step backwards to the animal cell again. These are both plant and animal cells are both eukaryotic cells, and eukaryotic cells mean that they generally belong to multicellular organisms. And additionally, inside the cell we have what are called organelles. So organelles are basically miniature organs within inside the cell that all play a specialized role, just the way that the organs in your organ systems in your body each play a specialized role. Right? So let me walk you through each of these as kind of an overview, and then we're going to talk about each of them individually. So let's start here with the boss's office, or the nucleus. The nucleus is a very large region. It's the largest quote-unquote organelle of the cell, and it is going to have chromatin inside of it, or a DNA. Now typically when you think of DNA, you think of that X, right? And that's going to be condensed DNA, which is DNA that you'll find just prior to cellular replication or cellular reproduction, mitosis, meiosis, for example. When we talk about that later, we'll talk about the condensing of the chromatin, which is how you see it here, all unwound, into those chromosomes. But generally, as you want to access the DNA to make RNA from it, it's going to be unwound and kind of loosely distributed like this throughout the cytoplasm. The very first thing that you'll notice, besides the chromatin, right, the DNA, is the nucleolus. And the nucleolus is the site of ribosome assembly. Ribosomes are going to be created here and secreted out through the nuclear pore. What else goes out through these nuclear pores, these little pink donut-shaped holes? Well, RNA does, right? So DNA is going to be the blueprint for, again, this is the entire recipe book of all the recipes. But RNA is just going to be a Xerox of a specific recipe. But every recipe is getting Xeroxed at once. So there's a whole flurry of different Xeroxes of these RNA molecules that are being created from different genes, from the chromatin. And the RNA is going to be allowed out of the nuclear pores. Now, if you imagine that there's gate guards here at the nuclear pores that are able to differentiate between RNA and DNA, they're going to do that a couple of ways. One, they're going to be able to tell what kind of sugar is there. So ribose would indicate RNA and be allowed to pass through. deoxyribose would not be allowed to pass through because it would be DNA. Additionally, RNA is typically single-stranded, and DNA is usually double-stranded. And there's a couple other reasons. Obviously, RNA is going to be much shorter than a long DNA molecule. But anyway, once the RNA molecule gets out through the nuclear pores in the nucleus, then it's going to reach what's called the endoplasmic reticulum, and that's shown here in pink. This is all part of the endomembrane system. Mind you, that sounds like a really big word, but it just means interconnected membrane system. And as we depict that here, you can see the nucleus touches directly to the rough endoplasmic reticulum, which touches directly to this region here, which is the smooth endoplasmic reticulum. But I digress. I mean, we have ribosomes, which are depicted as these little dots. The ribosomes are studding all of the rough ER. And the smooth ER does not have ribosomes on them, so ribosomes on the rough ER, no ribosomes on the smooth ER, and that's literally just what they saw under a microscope, where dots, so it looked rough, no dots, it looked smooth, and that's how they differentiated the two. But their major difference is that those ribosomes are the site of protein synthesis, so when the RNA leaves the nuclear pore, it goes to the ribosomes where proteins are created. After proteins are created in the endoplasmic reticulum, then they get transported to the specialized region called the Golgi apparatus, which is kind of like a mail sorting factory where the proteins are going to get little tags on them that identify where they're going to end up within the cell ultimately. And we'll discuss that when we get to the specifics of the Golgi apparatus. Other things that are depicted here is the mitochondria. Mitochondria, again, are very special. They are actually their own prokaryotic organism that got engulfed by a eukaryotic organism many, many eons ago, and they have a specialized external membrane that is specific to whatever type of cell that they're in. So if they're in a plant cell, it's got a specialized plant cell membrane. If it's in an animal cell, it's got the specialized animal cell membrane on the outside, and then on the inside, it has a specialized mitochondrial membrane. It also has mitochondrial DNA, which is completely separate from the DNA that's over here in the chromatin in the nucleus. And interestingly enough, you can trace this mitochondrial DNA back only through the maternal lineage, because unfortunately males, your sperm has the mitochondria in the neck, and only the head makes it in. So everything from the neck and tail down gets left outside of the egg during fertilization, which means that your mitochondria is not passed on. That means only mother's mitochondria is passed on to offspring. So you can track the maternal lineage of an individual through their mitochondrial DNA. We talked briefly about the smooth ER, but the purpose of the smooth ER is that it's going to help with the detoxification of the cell, and it also is going to create lipids, so it's going to synthesize lipids, which we're able to use in creation of things like the plasma membrane, for example. These centrioles you're not going to see again until we talk about cell reproduction, but these guys are going to be regions where we're able to create spindle fibers that are going to help bind to the DNA during the metaphase plate time of mitosis, and that'll get its own lecture a little bit later on, but that's when we'll talk more about the centrioles. We also have vesicles. Vesicular transport is basically the heart of this endomembrane system, so things that are going to be bound in vesicles that come off of the rough ER, for example, will get transferred to the Golgi apparatus. From one layer of the Golgi to the next, they'll get transported by vesicles, which are these little membrane-bound sacs that have things inside there. Right, but we also have specialized vesicles that have things that are going to be like digestive enzymes. So for example, this here is a lysosome that's going to be a cellular trash can or a recycling bin that's going to help digest proteins that may have been misfolded, etc. What else haven't I touched on? Oh, specialized type of regions like the flagella or the cilia that allow for the cells to have motion. And we'll talk about those individually as well, but a flagella would generally just be one on top of the cell, whereas cilia we're going to have multiple on the outside of the cell. All right, I'm going to move on to the plant cell, and I'm going to gloss over the stuff that I've already told you, like the nucleus and the nucleolus and the endoplasmic reticulum, rough and smooth, and the Golgi apparatus. And I'm going to focus on the new things that we haven't seen before. So, for example, here's the chloroplast. As I mentioned, it looked like a green stack of pennies, and this is where photosynthesis happens. This is where the cell is going to take energy from the sun and convert it into glucose that it can then use to be able to break down into energy later on. It can either be stored within the plant, or when the plant gets eaten by another animal or by an animal, it can be digested by the animal and then can be used for sugars that animal is able to use for its own energy source. Additionally, we have a central vacuole. A central vacuole is going to be where the plant stores water. It's also going to be where we put central pigments or any poisons that the plant might encounter. It will keep it away from the rest of the cell by keeping it in that central vacuole. And then we also have the cell wall, which is the major difference between the animal cells and the plant cells. Inside that cell wall, we have little holes. They're called plasmodesmata, and that's so that we're going to be able to allow water exchange and also information exchange from adjacent plant cells. Okay, So now that I've told you what I'm going to tell you, now I'm going to tell you. Um, so DNA is the hereditary blueprint. It is where all of the information is held within the cell. And it's a really long cookbook of all of the different recipes. Actually, it's a lot of different cookbooks if you consider that each chromosome would be considered its own cookbook. And there's a lot of different recipes in each one, and you could consider those recipes genes. And those genes are going to get transcribed into RNA. And that's basically going to be a Xerox of that recipe that's going to be used to guide the construction of the protein. In this case, the protein is going to be whatever food you're actually going to be eating. So while the RNA is vital and very important, you can't actually eat the recipe, right? The proteins are going to be what's doing the work in the cell. So the proteins are going to be the functional workers of the cell, and the RNA is going to be the blueprint that guides the creation of those proteins. Okay, so previously I mentioned unicellular organisms and multicellular organisms. And we're really not going to focus much on unicellular organisms for the purpose of this class. Those would be prokaryotic organisms. Prokaryotic means before the nucleus. And that would include things like bacteria, and those are going to, yeast, right? These are going to be the very simplest forms of life, single-celled organisms. Eukaryotic means that it has a true nucleus. Right? Prokaryotic cells do not have a nucleus. They still have organelles, but the organelles are all just kind of housed in the cytoplasm. And the eukaryotic cells are going to be cells that have the true nucleus and also have specific organelles that are going to have specific functions within the cell. And they're going to form things like animals and plants. We won't talk about fungi or protists for the purposes of this class. Now, as I mentioned, there's a ton of organelles, or miniature organs within the cell that have specific functions. They include things like the nucleus and the mitochondria, which I touched on just a minute ago, but I'll go into in depth in the further slides. It also includes the cytoskeleton, which I didn't touch on much, but I touched on it when we talked about the plasma membrane. And the cytoskeleton is the scaffolding that helps give shape, structure, organization to the cytoplasm and the plasma membrane of the eukaryotic cells. And if you picture it as scaffolding, you're actually kind of selling it short, because it's not just scaffolding, it's a meeting place for a lot of different proteins to congregate and interact with other biomolecules. So the cytoskeleton is also kind of like the town hall steps where people would meet and exchange information. It's kind of where the biomolecules might interact with one another at the cytoskeleton as well, is my point. Now this slide contains a lot of information. So I'm not going to go through all of them here, but what I would like you to look at are some of the specifics, like whether or not we have a cell wall. A cell wall has a function of protection and support, but it is not found in animals. Okay, it is found in plants. Now prokaryotes include things like bacteria and yeast, right? And so we do have a cell wall found there. Cilia and flagella are for motion, either for the cell to move through fluid, or that would be a flagella, or cilia would be more for fluid to move along the surface of the cell. And this would be something that would be absent in prokaryotes, because prokaryotes aren't multicellular, so they don't really need to move fluid across multiple cells. It's going to be absent in most plants and present in eukaryotic animals. So that's kind of what you're looking through as you look down here, is whether or not it's present in, or absent in prokaryotes, in eukaryotes, than plants that are eukaryotes, or animals that are eukaryotes. All right, and I'm going to talk about each of these and their function in the following slides, so I'm not going to linger too long on this table. Now, eukaryotic cells are multicellular. What multicellular organisms are comprised of, things like animals and plants. Again, we're not going to talk about protists or fungi, which would be mushrooms. And the diversity of eukaryotic cells is immense, right? If you think of all of the different organisms that are found on the face of the earth that are multicellular, there's a ton of them, right? So lots of diversity. And inside the eukaryotic cells, we have a cytoplasm. So the cytoplasm is going to be all of the aqueous portion, which is the cytosol, and all of the organelles. So it's going to house the nucleus and all of the organelles and mitochondria that perform those specialized functions within the cell. And again, we have specialized regions that are unique to both plant and animal cells. The major difference between plant and animal cells, just to hammer this home, is that we have a cell wall on the surface of plant cells. It also is going to cover fungi, some protists, but again, we're not really going to talk about those for the purposes of this class. So plant cell walls are going to have cellulose, which is a specialized polymer of sugar molecules that cannot be digested by most mammals, but it can be digested by termites, right? I told that bad joke in the previous chapter about the termites. And it's also comprised, plant cell walls are also comprised of polysaccharides, so sugars. Now, fungal cell walls are a little bit different. They're made of polysaccharides and a protein called chitin. But again, we're not going to really talk about fungi, so you certainly won't be tested on any fungal information. Plant cells also have something like that's called a plastid. I did not talk about that. That's a little storage region where it can store things like starches, etc., which is why when we eat plants, we can actually end up getting things like starch from them, for example, rice, or areas like the tuber, so like a potato would be a tuber and would have plastids inside of it. Also a central vacuole, which as I mentioned previously is a storage area for water, and additionally pigments perhaps, or any toxins that may have entered into the system. All of these are specialized organelles that are not found in animal cells. An additional organelle that is not found in animal cells is going to be chloroplast, but that'll get its own attention in a little bit. Animal cells are going to have vacuoles and cilia sometimes, but these are going to be separate from the type that's found in plant cells. So, for example, they might have flagella or cilia that plant cells do not have. Now, the plant cell walls are very porous that allows oxygen in and water in, etc., also carbon dioxide in. This is going to be to allow the cell to be able to have gas exchange and nutrient exchange while allowing it the structural support that it needs to be able to hold its own body weight upright in the absence of something like a skeleton, right, which is what we use to hold our body upright. So the plasma membrane is found just underneath the cell wall, and as I mentioned, there's little holes in it that allow the passage of biomolecules through. So the cytoskeleton is the scaffolding for the cell, and the cytoskeleton has three major types of fibers. The very smallest are microfilaments, they are the thinnest. The medium-sized ones are intermediate filaments, and the largest are the thick microtubules. And we will talk about microtubules when we talk about the cell division, mainly during mitosis. But this is just an overview of what I mean by that. So the microfilaments are these very smallest ones here. The intermediate filaments are depicted here, and the microtubules are depicted there. So they're a little bit larger, or they are larger, but they also play different roles. So as you can see, there is some overlap, but they actually are located to different regions. So the microfilaments are shown in blue, and the microtubules are shown in red. So they have different functions because they have different strengths and weaknesses. The microfilaments include something like actin, right? Actin is going to be involved in muscle cell contraction, and also is going to allow cells to migrate or change shape, and is involved during cell division for separating the two daughter cells from the parental cell. Intermediate filaments are typically twisted around each other like a ball of wire or ball of string, and they're involved in the internal scaffolding that helps maintain the cell shape. So it's going to be found right underneath the cell surface generally. And microtubules, including something like tubulin, are involved in cell transport of organelles and also of the cell transport itself, because they're the components of both cilia and flagella. And last but not least, again, we're going to see this mainly when we talk about cell division, it's involved in binding to the chromosomes and pulling the chromosomes from one side or from the center to either side during cell division. So the cytoskeleton is really important, right? It regulates multiple properties within the cell, including the shape of the cell, the motion of the cell, where the organelles move within the cell, and last but not least, cell division, which will get, again, its own chapter. And the microtubules are found inside cilia and flagella. And the most common example of flagella would be a sperm cell, right? When you have one long tail that propels the head of the cell through some sort of liquid media, right? Whereas cilia are multiple, so not just one, and they are generally found on cells that stay put, but are meant to propel liquids along them. Both of these are going to arise from a region that's a basal body, which is going to be comprised of those centrioles. So those centrioles that we talked about for cell division are going to have another purpose in terms of cell locomotion for cilia and flagella. So here's a basal body here, which is going to look just like those centrioles that we talked about, or we will talk about for cell division. And it's then going to extend out. These microtubule pairs are going to extend out through the entire long length of this tube. And they're able to contract and alternating, so the left side will contract and then the right side will contract, and that's going to allow it to give motion to the tail. This is an example of one cilium, multiple plural will be cilia, and this is the Paramecium, which is a single cell organism which is actually moving through a liquid media. So it's going to allow for this organism to move about and find food, etc. But generally, In a eukaryotic organism, cilia are on cells that are not going to be moving around at all, but instead are moving liquid across them. Now, if you were to compare cilia and flagella to, say, outboard motors or the oars on a rowboat, the cilia would be more along the lines of oars. There are multiple of them, they are on multiple sides, and they are all working in concert to create a motion of the liquid across the cell, whereas flagella are more along the lines of an outboard motor. A, they're much longer than cilia, which doesn't work with my analogy, but B, there's only one of them, and they propel the cell through the liquid environment. Now, while some unicellular organisms do use cilia to move through a liquid environment, mainly ciliated cells in eukaryotic cells are going to be found in places like the oviduct or the respiratory tract or the gills, and that's going to be moving water alongside the cells, which are going to be staying stationary. In animals, or eukaryotes, but animals specifically, the sperm we're going to be relying on one flagella for motion, and the major difference, of course, is that sperm are moving through a liquid interface, whereas ciliated cells are going to have liquid move across their surface. So this is another difference between the way the cilia work and the flagella work. So cilia work by, again, oars on a rowboat. There's a whole ton of them, and they're going to extend and then go all the way over and flip down, and then during the return stroke, they're going to bend so that you have less pressure, so they're not going to move as far, and then extend again and flip back down. And all of them together, see, these are cilia lining, say, the trachea, and we have mucus-secreting cells here, and so we want that mucus to move along so we can expectorate it or cough it up. And these cilia are moving in concert to move the liquid across the cell surface. Whereas a flagella, here's a human sperm, and the flagella from the human sperm is meant to propel the sperm through a liquid interface as opposed to having the liquid interface move across the surface of the cell. All right, so let's talk about the nucleus. This is a really good place to start in terms of talking about organelles and the organelle function. So the nucleus is the boss's office. It's the control center of the entire eukaryotic cell. It has three major parts. The first is that nuclear envelope. So if we talk about most of the plasma membrane being a lipid bilayer, this is actually a double lipid bilayer. So it's not just one membrane, it's actually two membranes just right next to each other. So it's a double lipid bilayer that has these nuclear pores, and those were depicted previously as little pink donut rings, right? And those nuclear pores are going to allow certain things out and not other things. Additionally, inside the nucleus, we find chromatin. Chromatin is generally going to be decondensed and kind of sprawled everywhere throughout the nucleus. But during cell division or just prior to cell division, it's going to get condensed into chromosomes, which is what you're familiar with in terms of like those X's that you see when you think of chromosomes. But chromatin is DNA. It's just unwound DNA, and it's accessible DNA that's going to be able to be used to be able to make those copies that I was talking about in terms of RNA. And additionally, we have that really dark central region called the nucleolus. And the nucleolus is often glossed over in the nucleus, but the nucleolus is very important because it is the site of ribosome assembly. And ribosomes, which are made of RNA, by the way, ribosomal RNA, are actually going to be very important in creating proteins, right? So the nuclear envelope is very important because it allows certain things to come through and other things not to. It's a selective exchange of materials, more selective than, say, the regular membrane. And the nuclear envelope is a double membrane perforated by what we call those nuclear pores. And those nuclear pores allow certain things in and out, no problem. Water, ions, small molecules that might be needed inside, no problem. But larger things like proteins, ribosomes, RNA, DNA, which is not allowed out, are regulated by these specialized gatekeeper proteins that are called the nuclear pore complex. And they look like this, right? So this is what they look like in our pretty little diagram. This is what they look like in an actual photograph. So this is a pseudo-colored, or they basically added the color afterwards to a black and white image that's from a scanning electron microscope. And I know that because a scanning electron microscope is more three-dimensional and a transmission electron microscope is more like slices, so it'd be two-dimensional. And you can see here these are nuclear pores that are lined with those nuclear pore complex here of a yeast cell. Now if you were to take a cross-section again, you would find that unwound chromatin in the center, and the nucleolus, which again would be the site of ribosome synthesis. So chromatin is DNA that is unwound. It's found in the nucleus, and it's generally going to be kind of tossed all over the place because it's constantly being used to create RNA. But prior to cell division, You kind of have to clean house a little bit. So I like to say that if we were to, instead of using a recipe book, let's just suggest that all of the old recipes are on these scrolls that are very long and contain a lot of recipes on them. So instead of books, they're scrolls. All right. So if you're using the scrolls in the library to make a bunch of Xeroxes for everything, they might be kind of tossed all over the tables, for example. That would be chromatin, kind of unwound and kind of tossed around everywhere, accessible, but not necessarily pretty or packaged. But during cell division, this is when you have to clean up very quickly because the mother-in-law is coming over or something, or someone's coming to check the library, or actually in the case of cell division, you're creating a whole new library with copies of the scrolls. But I digress. My point is that you would want to clean up a little bit. You would want to condense everything and put them away. And chromosomes are the cleaned up, condensed version of DNA. Now, they are inaccessible at this point, though, so keep in mind that during cell division, you're not able to make RNA from the DNA because the really tightly coiled things that we think of as those X-shaped chromosomes are bound so tightly that we're unable to access any of the DNA during that time frame. So chromatin is the accessible version of DNA. Now, chromosomes contain genes, and if you picture the chromosomes as a recipe book with a lot of different recipes, genes are the recipes themselves, and the recipes are going to provide a blueprint for a bunch of different proteins, right? And each protein is going to have a different function, the way that every food might have a different flavor. Now, proteins, again, they're synthesized in the cytoplasm. They're made at the site of the ribosomes. Ribosomes are found either free in solution or bound to the rough endoplasmic reticulum. So in order for them to get to the point where they're going to be able to be created, they have to first leave the nucleus. All right, they have to do that going through the nuclear membrane. They'll go through those nuclear pores. But because DNA is not allowed out, we have to make that Xerox of it first. And that Xerox in this case is going to be messenger RNA. So the messenger RNA is a copy of only one gene. It's not going to be a copy of multiple genes or the entire chromosome. It's just going to be a copy of the recipe that we need to make whatever it is that we're trying to make, our cherry cobbler, right? And that recipe then is going to travel, the Xerox of that recipe is going to travel through the nuclear pores into the cytoplasm, where ribosomes are going to meet up with the messenger RNA and direct the synthesis of whatever protein is found in that recipe. Okay, so just to kind of show you a couple things here, these are very obviously plant cells. How do I know? Well, these look like bricks, right? That's a cell wall. Now, what's the nucleus? This here is the nucleus. And normally, the nucleus houses what we call chromatin, which is decondensed DNA. And that looks kind of like this. These are the scrolls tossed all over the library. But this is when we're cleaning everything up and all the scrolls are tightly coiled and put back in their place. And those are called chromosomes. And again, this is going to be only something that is visible during mitosis, or when meiosis, I suppose, when we are creating copies of the cells, so for during cell division. Again, inside the nucleus, we have another specialized region called the nucleolus, and it's often glossed over, but it's very important because it's where ribosomes are made, and ribosomes are very important because ribosomes direct protein synthesis, right? So inside the nucleolus, we're going to be making ribosomal RNA and then different proteins that are going to help create and assist with that. Ribosomes that are in various stages of synthesis, so different partially created ribosomes, and also DNA. All right, so what do ribosomes look like? So there's a lot to take in here, so let me walk you through what you see before we walk you through what's happening. So first, we're looking at an mRNA molecule, and that is shown here, a messenger RNA molecule, and that is shown here in red. The messenger RNA is being surrounded by ribosomes. Ribosomes are also RNA molecules. We have a small ribosomal RNA, large ribosomal RNA. These subunits come together and they're going to read the messenger RNA in something called codons, which we'll talk about later, but in small subunits of the mRNA, they read it, and every time that they read it, they add an amino acid, which then becomes a long-growing protein dangling off of the ribosome. Okay, and that kind of looks like this. If you look at it under a transmission electron microscope, the ribosomes have a small and a large subunit, and the proteins that are dangling off of that, each one of them, is, as it's getting further along the chain, it's getting a longer and longer polypeptide that's growing. Okay, so I used the word endomembrane system previously, and again, it sounds like a pretty large word, but it's really very simple. What it means is connected membranes. And that's going to mean that we, just like your house has different rooms, but all of the rooms are connected by the same walls, the nucleus and all of the other organelles, although they are separated from each other by plasma membrane, that plasma membrane is connected throughout the entire series of organelles. And that helps that both segregate and ensure that the orderly occurrences of these biochemical processes happen in a specialized, regular fashion. So the different organelles involved in the endomembrane system include the nuclear envelope, so not the nucleus itself, but the nuclear envelope, which is attached directly to the rough ER, the rough endoplasmic reticulum. The rough ER is attached directly to the smooth ER, and then is also connected to the Golgi apparatus by way of vesicular blebbing. So vesicles are going to bleb off of the ER and then get picked up by the entry face of the Golgi apparatus, travel through the Golgi apparatus, also by vesicular secretion and absorption until they get to the exit face where they've got a bunch of tags on them that say specifically where they're going to end up going. Another thing that's part of the endomembrane system is everything that's a vesicle. So again, the vesicles bud off of the endoplasmic reticulum and go to the Golgi. They bud off of the Golgi and go to wherever their end location is. We also have vesicles that are lysosomes. And lysosomes are going to be responsible for breaking down misfolded proteins. We also have something called a peroxisome, which creates peroxide and helps break things down. And these guys are going to be the cellular, both trash can and recycling center. So it's going to break molecules that we no longer need or we consider to be invasive, like something like a bacteria that we've caught and eaten and degraded, and into their smallest biological molecule that we can use or discard. And then the lysosomes will then recycle what we can, and whatever we cannot, it will bind to the plasma membrane and release to be picked up by the bloodstream. All right, so we talked about vesicles, but I didn't talk about what vesicles are specifically. So vesicles are these little membranous sacs that have the membrane around them and different biomolecules inside of them. It's basically a transportation system or a bus system whereby proteins get to various regions of the membrane system. And they do that by traveling again through these vesicles. And exocytosis is when things actually are going to leave the cell and export the contents outside of the cell. Additionally, the cell is going to undergo endocytosis, so endo being enter, exo being exit, so exiting the cell, entering the cell. Endocytosis is when we are bringing things in by way of the plasma membrane, extending around whatever biomolecules we want, and pinching off and fusing a vesicle inside the cell. And these two are equal and opposite processes that are happening the entire time, because some proteins are just meant to be exported. They're not meant to be kept inside the cell. Now, the very first place that the protein, well, where the proteins are going to be created, but the very first place of the endomembrane system after we leave the nucleus is the endoplasmic reticulum. And the endoplasmic reticulum is basically a long series of membrane enclosed channels that's found within the cytoplasm that houses ribosomes. And it's a labyrinth that has a ton of ribosomes. And what are ribosomes for? Ribosomes are for protein synthesis, right? So the rough ER is the site of protein synthesis, but it's not the only type of ER, right? We have a second type of endoplasmic reticulum, and that's the smooth endoplasmic reticulum, and that's where all phospholipids are created. So rough ER creates proteins, smooth ER creates phospholipids. It also helps detoxify the cell a little bit. And this is what I meant when I said they labeled it based on what they saw, right? In the early stages of scientific research and discovery, you either labeled it by what it looked like or you labeled it by your last name. And you'll see that a lot throughout the course of these series of lectures as well. So again, the rough ER is studded with ribosomes, so it looks like it's a lot rougher of a ride. Here's a smooth ER. It's nice and smooth. These highways don't have any bumps on them, so that's why they called it the smooth ER. Now, the rough ER, again, these ribosomes, site of protein synthesis. I cannot hammer that home enough. And the proteins that are created then get caught in the little blebs off of this endomembrane system and go to the Golgi apparatus. The smooth ER is the area where we are creating our, we are detoxifying, and we're also creating all of our phospholipids, which are going to basically be used to create all of the membranes and also this endomembrane system. Right? Rough ER, studded with ribosomes, creates proteins. Smooth ER, no ribosomes, detoxifies drugs and synthesizes lipids, hormones, etc., and again, phospholipids that are going to be involved in creating the plasma membrane. So let's move on to the Golgi apparatus. So the Golgi apparatus is basically like the postal service or the packaging system for proteins. So once proteins are made, they're basically baby proteins, and they are going to come out of the endoplasmic reticulum into little blebs that are going to meet up with the Golgi apparatus. And just like if you picture that you had to sort mail, and you got mail from a little region somewhere in the United States, and first you had to decide whether that envelope that you have in your hand is going to go north or if it's going to go south. And then you have to decide if it's going to go east or if it's going to go west. And then once you finally decide east, west, north, south, you have to decide which state it's going to go in that general vicinity. Then you have to decide which county. Then you have to decide which neighborhood and which streets and which house specifically. And so as the proteins go through the different levels of the Golgi apparatus, they get different modifications or different tags on them such as carbohydrate groups, etc. Some things are going to get modified, making smaller proteins or larger proteins, etc., that are going to give it specific tags that are specific labels for its end destination within the cell. And when it leaves the Golgi apparatus, having gone through level A, B, C, D, E, etc., it's going to end up in a vesicle where it's going to end up being secreted and end up headed to its final destination. So the purpose of the Golgi apparatus is to separate the proteins that were created from the ER. It also accepts the lipids, right, because the lipids are going to be part of that vesicle. Then it's going to help sort them out according to their destinations. And then the molecules are going to be packaged into vesicles that are then transported to wherever they're going to, again, end up at their final destination. So as they come into the Golgi apparatus, on the entry face, they have no labels. This is when we're deciding north, south. And then from there, those blebs go next, and then you decide east, west. And then we decide from there, state. Then we decide again, neighborhood, and then street, and then finally house. And everything that's going on this particular vesicle, on this bus alone, is going to the same location. In that way, we can kind of save time, and the endomembrane transport system can get the proteins to wherever they are destined to go to. Right? So proteins are modified as they move through the cell. They're basically made in the rough ER. They travel through multiple layers of the Golgi where they have tags added to them, and then they're modified, and then they're exported or kept within the cell if they're meant to. But if they're meant for exportation, they're exported through the plasma membrane. We also have specialized proteins like antibodies and glycoproteins. These are specialized, and they're created by and utilized by white blood cells, and they attack foreign invaders and basically alert the body that this is non-self and activate the immune system so that these guys can end up targeted for destruction. All right, so what does that actually look like? So here's an antibody that's going to be created just like all proteins, because antibodies are proteins. It's going to be synthesized on the ribosomes here, depicted here on the rough ER, and then transported through vesicles or vesicular transport to the Golgi apparatus. It hits the entry face of the Golgi, goes through multiple blebs, goes through multiple different compartments until it ends up on the exit face of the Golgi, where it's in a vesicle that's destined for exocytosis or release into the interstitial fluid, and then it'll get picked up to the bloodstream where it'll circulate around and be able to help attack any foreign invaders that it finds. So I talked about lysosomes when we talked about the endomembrane system. It's a specialized type of vesicle, and it says digestive system, but it's more than that. It's also a recycling center. So there's a lot of digestive proteins in there which are going to break biomolecules down into their very smallest components. And anything that can get recycled is going to get recycled. But anything that can't get recycled is going to get sent out for export outside of the cell for release into the bloodstream like cellular waste. So basically, when the cell runs into something exogenous, like a foreign invader, we might turn it into food, right? We might be able to grab it and pull it into the cell and start to digest it. So the lysosome's job would be to fuse with that food vacuole and digest the food into the basic nutrients, and then again decide whether or not we're going to be able to recycle those nutrients, or if we're going to have to release them out into the extracellular fluid. So that looks kind of like this. The cell intercepts something on the external environment that it's considered to be food, right? So it pulls in that food vacuole, and that food vacuole is going to bind with the lysosome vacuole, right? The lysosome vacuole is going to have enzymes in it. Where do those enzymes come from? Well, if we start all the way back at the beginning, the enzymes, just like all proteins, are synthesized by ribosomes, and they go into vesicles, and then they go through the Golgi. The Golgi gives them lots of tags, and eventually they end up destined to be a lysosome. So the lysosome is then going to interact with the vesicle that has the food vacuole. Now these two come together, turn into kind of like a peanut, and it's going to digest all of the food and decide which molecules are going to be able to be recycled and reused, and which ones are going to have to be expelled back out as waste into the interstitial fluid. All right, but we also have specialized type of vacuoles in certain organisms, like freshwater organisms, very small single cell organisms, have special contractile vacuoles that are composed of collecting ducts, a central reservoir, and then a tube that comes in, and that allows the excess water out of the organism, and they are able to use that. Generally, they use it to pump salts out of their cytoplasm, but they're also able to contract, which squirts the water out of a pore in the plasma membrane, and when that water expels this direction, just like Wally in the fire extinguisher, the organism goes that direction. So it allows them to actually move around in an emergency, for example, if they see a predator or whatnot. And the organism in this case that we're discussing is something called a Paramecium, which is, again, a single-cell organism. But generally, when we talk about eukaryotic cells, we're only going to see central vacuoles or any vacuoles in plant cells. Central vacuoles are going to occupy a lot of the space in the plant cell, and they provide what's called turgor pressure. So anytime you see the plant that has begun to wilt, for example, it's kind of fallen all the way down to the ground, it's no longer standing up properly. And then you add a little bit of water, or maybe a lot of water, to the soil, and the water gets pulled up through the root system back into the cells of the plants. And then the plant starts to perk back up and eventually stand back up on its own. And what you're seeing there is turgor pressure. Turgor pressure is literally the water pressure, the pressure within the cell itself. In this case, it's stored inside that central vacuole, and that central vacuole is going to push out all the organelles of the cell, which is going to provide the rigidity that the cells need to be able to have the plant standing upward. Multiple functions of the central vacuole, although first and foremost it's meant to maintain the water balance, right? It's a water storage area, and it's going to be able to use that water to provide cellular resource for when the cell needs it in metabolism. But it also is going to be an area where the plant can store nutrients or pigments or hazardous waste that it might want to keep separate from the rest of the cell. And last but not least, to be able to provide turgor pressure or water pressure to keep the cells rigid. All right, so moving forward, we're going to talk about mitochondria and chloroplasts. Mitochondria and chloroplasts are both very special in terms of organelles. And that's because, unlike the other organelles, mitochondria and chloroplasts were originally their own organisms. That hypothesis is called the endosymbiont hypothesis. And basically what they believe is that mitochondria and chloroplasts were originally prokaryotic bacteria, but they had specialized function, right? Chloroplasts undergo photosynthesis. Mitochondria create energy through cellular respiration. And so when another organism engulfed that prokaryotic cell, instead of digesting the chloroplast or the mitochondria, it became apparent to that organism that it could keep that prokaryotic cell alive and use it to harvest energy. All right, so mitochondria, again, they're involved in cellular respiration. Basically what they do is they take sugar and break it down and produce ATP. And there's a lot of steps in this, and if you want a chapter on photosynthesis, it's not going to be part of my 101 class this semester, but I'm sure you can find many resources for this online. But basically what happens in photosynthesis is that chloroplasts are going to take the energy from the sun and use it to create sugars. Right? So those sugar molecules are stored in the cells of plants and then can either be used to drive biological reactions for those cells themselves, or they can be used to drive biological reactions for animals that eat those plants. Right? Because animals aren't able to create their own sugar, so they have to get their sugars from somewhere else. And the primary source of all sugars comes from the energy from sunlight being converted into sugar molecules by photosynthesis inside chloroplasts. All right, so these single-celled prokaryotic organisms, which eventually became eukaryotic organelles, have a very specialized function that the cells cannot provide on their own. So while they are considered to be organelles, they actually have a very special history compared to some of the other organelles. And how do we support the idea that they were originally their own prokaryotic cells? Well, one, they're very small, about 1 to 5 micrometers in diameter, on the smaller size of any of the cells, definitely in the prokaryotic cell range. And they have a double membrane. And what's interesting about the double membrane is it's kind of like a way that the mitochondria and the chloroplasts hide within the host cell. So the outer cell, or the outer membrane, is going to mimic that of the host cell. So not just a mammal cell, but a dog cell if it's in a dog, or a human if it's in a human. But the inner membrane is going to be the same for everyone. It's a mitochondrial membrane or a chloroplast membrane. So they have a specialized membrane that they keep for themselves, and then an external membrane that they use to hide within their host. Additionally, they synthesize their own ATP. That means that they create their own energy. So previous to being inside this other organism, they would have been able to survive on their own. But the eukaryotic cell does offer great protection, which is one of the reasons why it's believed that this is a working relationship, basically. Both chloroplasts and mitochondria have DNA and ribosomes, so they have the ability to reproduce on their own. Again, mitochondrial DNA is specialty to the maternal lineage, and again, chloroplasts also have their own DNA, so they're able to create their own proteins and drive their own cellular processes independently of the host cell itself. So these are basically like renters that take up space and pay their rent and just kind of keep hidden within the cell, and in return they get protection from the eukaryotic cell. Okay, so mitochondria are involved in energy extraction. They take sugar and they break it down into the high energy bonds of they break down, sugar, and then they create ATP, which is where energy is stored and then is able to travel throughout the cell to be able to provide energy to any number of metabolic processes. And we are able to take the energy from a glucose molecule and extract it in two different ways. We can do it with oxygen and without oxygen. Now, under anaerobic conditions, which is without oxygen, we yield a very small amount of ATP. However, we do yield ATP, so it is energy, but it's nowhere near the amount of energy that's yielded under aerobic conditions. So the anaerobic conditions, I believe we get 4 ATP per glucose, and aerobic gets us up to 36, so it adds another 32. So it's a little bit less than tenfold the amount of ATP that you can get from the same glucose molecule when you have oxygen in the equation than when you do it without oxygen. All right, inside the mitochondria, again, we have that inner mitochondrial membrane, which is specific to the mitochondria itself, as opposed to the external membrane, which is going to mimic the host membrane. That inner membrane is folded into these linings that we call cristae. And the cristae is going to be the site of what we call the electron transport chain, where we are going to be extracting more energy from the glucose molecules. And we have a central region called the intermembrane compartment that's going to lie between that inner and outer membrane. And there's a matrix space within that inner membrane, which is going to allow for the specific structure of this is what allows for electron transport chain to take place. And don't worry, I'm not going to go into the specifics of the electron transport chain here. So this is what I mean when I say it hides inside a membrane of the host cell. So this outer membrane is going to appear to be whatever the organism that its host is on the external surface. On the inner membrane, however, it's going to be a mitochondrial specific membrane. So all mitochondria across all organisms are going to have very similar inner membrane structure. We also have this intermembrane space, which is found in between the inner and the outer membrane. And the cristae are these folds that are going to be found in the intermembrane, in the inner membrane. And this intermembrane space is going to have a specific fluid, and the inner membrane space has a different fluid known as the matrix. And as things are going to travel across that, that's what's going to allow for the electron transport chain to take place. This is a cross-section of a mitochondria. This is a transmission electron microscopic image, and you can see these folds here are the cristae. Inside here we have the matrix, and then the intermembrane space is found in between the inner membrane and the outer membrane. All right, so chloroplasts, again, these are very specialized organelles. They have a double membrane surrounding them as well, so similar to what we just saw with the mitochondria. And again, that outer membrane is going to appear to be whatever the host cell is, and the inner membrane is going to be chloroplast specific. And that is going to be a region within that inner membrane that holds these membranous sacs that are called grana. And each grana is made of an individual molecule called an individual thylakoid. All right, and a thylakoid is like a coin, basically. It's a coin-shaped region, and a stack of those coins is called a granum, so a stack of thylakoids. And this is going to be how photosynthesis takes place. And again, I'm going to spare you all of the details of photosynthesis, but this is where all the magic happens. This is where sunlight is going to be converted into sugars that the plant is going to be able to store and then either use later on, or again can be basically the beginning of the food chain. And if we look at a TEM image, so a transmission electron microscopic image, you can see these granum or stack of thylakoids found within the chloroplast. Inside that thylakoid membrane, we have a green pigment. It's called chlorophyll. More like borophyll, right, if you've ever watched Adam Sandler. But chlorophyll is a specific pigment, and there are many other pigments, but chlorophyll is a specific pigment that captures sunlight in a specific range. Again, the other accessory pigments capture sunlight in other wavelengths, and that's how it's going to be able to capture that energy to create sugar from carbon dioxide and water. And really the very basis of what I want you to take home here, well, multiple things, but one of my major points here is that plants are taking carbon dioxide and using carbon dioxide and water to drive their reactions, and they are off-gassing oxygen. And that's how they become carbon sinks. They use that to create glucose, and glucose is a chain. They chain that up into cellulose, so that actually literally the entire mass of the tree comes out of thin air. It comes from the creation of sugar molecules from water and carbon dioxide gas. And this is why carbon sinks are so important. Places like the Amazon, which is undergoing deforestation, etc. It's releasing carbon back into the atmosphere, which is disrupting the carbon cycle. And plants are the exact equal and opposite. This is the point I'm trying to hammer home. Plants are the equal and opposite set of reactions to animals, because as animals, we inhale oxygen and off-gas carbon dioxide. And so we truly need plants in order to create and return the oxygen back to our air so that we can breathe it, right? So it's a cycle. All right, but I digress a little bit. I apologize. Let's talk again about different regions within the plant cell that are specialized that we don't see in animal cells. Another organelle that we find only in plants are plastids. And plastids also have a double membrane and are storage containers. This is where we're going to store, well, pigments, but starches is what we're mainly going to be concerned with here. So we're going to store them in things like a rice granule, or a potato, which is a tuber, which is another place where a plant might store starch. And so plastids look kind of like this. They're little vacuoles that store starch molecules. They look like that, little globules. And if you end up with an area like a tuber, we're going to have a lot more plastids in those regions than they do in other parts of the plant. Okay, so we just talked a lot about eukaryotic cells and all of the different organelles. And now I'm going to digress a little bit, and we'll talk about prokaryotic cells. Prokaryotic cells are much smaller than eukaryotic cells. I think I've hammered that home a couple times. And while they do have specialized surface features in some ways, they also have fewer specialized structures inside the cytoplasm themselves. So you can kind of compare this to the type of house that someone lives in, right? So a eukaryotic cell is kind of like a large house with multiple different rooms that have specialized function. So you have a separate room in which you might bathe, from a room in which you might sleep, from a separate room in which you might cook, from a separate room in which you might lounge, right? And a prokaryotic cell is more like a hut. People who live inside a hut still have to bathe, still have to eat, still have to sleep, but they might not have specialized rooms. They don't have specialized rooms within which this occurs. It all occurs in the same area, and that's kind of how prokaryotic cells are. Inside the cytoplasm, a lot of the same things still happen. DNA still makes RNA, RNA still makes protein, but they lack a nucleus. So there's not a separate region for DNA to make RNA, then there's a separate region for RNA to make protein, which is how it works in eukaryotic cells. So without these specialized regions within their cytoplasm, all of the same functions that are still occurring are occurring in the same location and kind of all at once. Additionally, they're just, in general, they're less complex, right? They have a more simple internal structure. They have more simple DNA, although they do have DNA. They're generally much smaller, approximately 5 micrometers long. They are going to just divide and make an exact copy of themselves. Oftentimes, they have a stiff cell wall that surrounds them that's going to offer protection. And if you know anything about bacteriology, right, or microbiology, then you will know that bacteria take several different shapes. So not all bacteria look the same, and they also don't all respond to the same antibiotics or course of treatment, right? So some of them are rod-shaped. Those are going to be something like a bacilli. They're going to look like your E. coli, for example, are rod-shaped. We also have spiral-shaped spirilla. These are going to help embed in the walls of the cells that they, or in the cell membranes of the cells that they're invading. Also spherical bacteria are going to be called cocci, like streptococcus, for example. And so this is just to show you what we're looking at. So this one here is going to be a rod-shaped bacteria. This is a bacillus prokaryotic cell. Obviously, all these are prokaryotic cells. The outside is going to have this capsule around it. That's not always present, but it is often present. Additionally, a cell wall, again, not always present, but usually present. And then underneath that, we have the cell membrane. Inside the cell, all of the same things are happening, right? We have ribosomes that are going to be the site of protein synthesis. We have food granules that have been brought in by endocytosis and are going to be digested. We are going to have the DNA, right? In this case, it's a chromosome. We don't have a nucleus, but we have a region that's like a nucleoid region, the same way you might not have a bedroom inside a hut, but you might always sleep in the same place. So this is going to be the nucleoid region where the nucleus is present. But what's interesting about prokaryotic cells is that DNA is going to be used to make RNA at this exact same location that RNA is going to be used to make protein. And sometimes these structures are actually going to be tied to one another. So while the DNA is actually being copied into RNA, the RNA is making protein in the exact same time frame. And that's very unique from a eukaryotic cell where these are housed in separate compartments entirely. Additionally, some of these prokaryotic cells actually are able to photosynthesize, and they do that in a similar but yet different way from chloroplasts. Here we have a photosynthetic prokaryote, and we have these photosynthetic membranes that are going to lay on top of one another, basically like rings in a tree or like a rolled up layer of dough. And as it crosses through each one of these, it's going to serve the same process in the photosynthetic capabilities that the thylakoid stack did, or the stack of thylakoids, the granum, did in the chloroplast. Now, bacteria have a specialized type of polysaccharide coating that they're able to secrete. We have slime layers and capsules. Now, capsules and slime layers are very similar. Slime layers actually start off, and then slime layers end up hardening into capsules or biofilms. And you can kind of picture the difference between plaque and tartar, right? So capsules are going to be much harder to remove. So capsules are going to be the hardened version of a slime layer, and this is going to help form films of bacteria on surfaces, for example, your teeth, for example, or urinary tract infections. If they get a capsule around themselves, it's going to make it very difficult for antibiotics or even antibodies to locate and destroy these foreign invaders. So they protect the bacteria, and additionally, in a non-aqueous environment, it also is going to help keep them from drying out, and desiccation or drying out would obviously kill the bacteria. So on top of that, they have these external surface hairs. They're called pili. And pili have two major functions. The first that is going to assist in what I was just talking about, which would be invasion, right, and infection. And they have attachment pili, is what I was referring to. And these are very short, they're very abundant, there's a whole lot of them, and they help the bacteria basically stick to the surface of whatever cell that in the tissue that they've infected. But the other thing that's dangerous about bacteria is they actually have exchange of DNA from one to another through what are called sex pili. Although they don't actually have sexual reproduction, they have exchange of genetic material. And this is what helps contribute to the recent increase in antibiotic resistance, for example. So if an individual fails to take the entire course of their antibiotics, and a few of the bacteria that were weakened by but not killed by the antibiotic survive and end up with a specialized type of gene by natural selection. So the ones that had that gene were going to survive more than the ones that didn't. And then they're able to pass the genes that are beneficial on by conjugating with the bacteria that doesn't have that gene through these sex pili. So this can be very dangerous, and is how antibiotic resistance can spread through a bacterial colony very quickly. All right, inside the prokaryotic cell, we have a region that's similar to the nucleus, but it's not a nucleus because there's no membrane surrounding it. It's called the nucleoid region, and it is going to be a little bit separate from the rest of the cytoplasm, and it has a circular chromosome of DNA. So instead of having those X's, it has a circle, and there's also specialized regions of DNA that are called plasmids, which are smaller rings of DNA found in the cytoplasm. And scientists actually use plasmids to our advantage. For example, if we want to create insulin, we can pop in a plasmid that has the gene for insulin into bacteria. We can make that bacteria, use that bacteria basically to make insulin, and then we can lyse all those bacteria open and harvest the insulin and then give it to diabetics at a reduced cost than having to create it somehow in their labs. So we oftentimes will use bacteria to create whatever protein or gene of interest that we would like simply by popping in a plasmid that has the transcript that we're interested in. But I digress. As I just mentioned, prokaryotic cells have no nuclear membrane, so they have a nucleoid region but no nucleus. And they have no membrane-bound organelles, but they do have specialized membranes, some of them, that are able to capture light and undergo basic photosynthesis. And they're able to do a lot of the same things that eukaryotic cells are able to do, right? They digest food, they are going to have ribosomes, which is a site of protein synthesis, so similar to what happens in eukaryotic cells. So there's a lot of similarities between prokaryotes and eukaryotes, but generally the major differences are that the eukaryotic cells are going to be multicellular organisms and have specific organelles within them that have specialized function. Thank you so much for listening to me today. I really appreciate your time. Happy studying, and aloha.